I'm going to move on to Evangelos Kiskinis, who's going to give us our next presentation. He is an assistant professor of neurology and physiology at Northwestern, has made several novel important insights into ALS biology, in particular nuclear cytoplasmic shuttling and the nonsense-mediated decay pathways. And so I'm going to turn it over to Evangelos.
Okay, thank you, Bob. Let me just share my screen. Okay, so hopefully you can see my my presentation at this point. And so thank you for that kind introduction, Bob, and hello to everyone. Thank you for tuning in. And thank you for putting together such a wonderful program for this year's meeting. I'm delighted to be here. And although it's unfortunate that we cannot be together as a community, this is the next best thing. And I'm looking forward to the rest of the day. I also want to take this opportunity to say a big thank you to the Les Turner ALS Foundation that have supported my lab over the last few years. And their seed funding has been crucial for the establishment and growth of my group. And for that, I'm really grateful. So what I'd like to do over the next 20 minutes or so is give you an update on our efforts to develop models of ALS using reprogramming approaches, and our focus on understanding the mechanisms that drive the disease. See how I can turn on the slides. Okay. Very briefly, ALS is a devastating neurodegenerative disease that's characterized by a gradual inability to control movement that leads to progressive paralysis and eventual death. And the clinical manifestation of the disease is driven by the relatively selective degeneration of motor neurons, which are found in the brain, brainstem, and spinal cord. These are very specialized neuronal subtypes because they sit in the CNS and send their long axons outside the central nervous system to synapse with muscle and control our movement. And while there's been tremendous progress, particularly in understanding some of the genetic causes of the disease, as well as the cellular mechanisms that drive the dysfunction and the degeneration of these motor neurons, effective treatments have been lacking. And we and others have suggested that the technical inability to access and study these human motor neurons from patients has really hampered the goal of developing therapeutics. Now, to override this lack of access to the central nervous system, we, as well as others, have been using reprogramming approaches. And this is a simplified schematic of the general strategy that we take in our lab, which is based on the generation of induced pluripotent stem cells. So we typically identify a patient that we want to study. We isolate some sort of somatic cell type from this individual. This used to be a skin fibroblast. Now it is blood cells. And we use a cocktail of molecular factor to turn these somatic cells into what we refer to as patient-specific induced pluripotent stem cells. The advantage of these cells is that they divide indefinite, so we can make loads and loads and loads of them, and they retain the ability to be differentiated into, excuse me, any cell type in the human body, including motor neurons, which are the cells that are become defective in this disease. And once we have these cells in culture, we employ a range of genomic, functional, and biochemical analysis, comparing them to controls to identify pathways that become defective. We can apply patient-specific drugs. And eventually, we always want to do two things. We want to assess the relevance of our findings in the culture dish by using in vivo model systems. In the past, we've used flies. We're going to talk about some exciting worm models today. And we also eventually want to apply what we learned to the systems back to the patients, either by looking at postmortem tissue or other approaches. The last thing that I wanted to mention is that the advent of next generation gene editing technologies has really allowed us to manipulate the genome in these cells effectively. And in cases where we know of a genetic insult that's associated with a particular type of ALS, we can use CRISPR-Cas9 to correct mutations, or in other cases, we can introduce mutations in a wild-type genetic background to create pairs of isogenic control lines. And I'll show two examples of these strategy today. You know, this has been, as I mentioned, there has been tremendous breakthroughs in ALS genetics over the last decade or so, with loads and loads of new genes having been identified. And in fact, at this point, we know of Mendelian variants in more than 25 genes, as well as of multiple genes that modulate susceptibility to this disease. Now, however, the overwhelming majority of patients are diagnosed with sporadic ALS, while a small percentage ranging between 10 to 12% are clearly diagnosed with clear familial disease, which is likely driven by genetic causes with high penetrance. And we heard about some of these genes earlier today. C9orf72 is by far the biggest genetic contributor to ALS, and others like SOD1, NEK1, TDP43, and FUS are all major genetic contributors. Now, at face value, the fascinating observation about ALS genetics is that it appears to be quite heterogeneous. In other words, these genes encode for proteins that seemingly do very, very different things. However, the discovery of more ALS genes over the last few years has really begun to illuminate an emerging pattern of cellular pathways that, when they become perturbed, can lead to motor neuron dysfunction and degeneration. And I've done what others have done before here, which is categorized the genes that cause ALS into three broad pathways. The first one is RNA metabolism. We just had a beautiful talk from Magda about how TDP43 and FUS mutations lead to dysfunction of this pathway. Other genes in this group include matrin3 and synataxin. The second broad group of genes include things like C9orf72, VCP, ubiquitinin, which are involved broadly in proteostasis, and the lysosomal function, autophagy, and so on. And the last broad group is of genes that are involved in cytoskeletal homeostasis and trafficking, transport of organelles within neurons. These include things like NEK1, kinesin 5A, tubulin, and so on and so forth. And however, it's important to state that there are genes like SOD1 which do not fit, at least seemingly, any of these pathways. So our eventual goal is to develop iPSC-based models for each one of these genetic factors. And the fundamental question we're trying to address is whether these share mechanisms that lead to dysfunction of motor neurons and eventual degeneration. And a major focus in our lab over the last few years has been C9orf72. And early last year, we published a study describing the devastating effects of this repeat expansion mutation in the subcellular localization of proteins, highlighting a shift in the balance between protein translation and RNA degradation through this pathway known as nonsense-mediated decay. And I'd also like to mention that we co-authored a study which was published last week, which was led by our close collaborator, Uday Pandey at the University of Pittsburgh, highlighting a novel functional interaction between two ALS causal genes, matrin3 and C9orf72. And we have projects focusing on NEK1 and preliminary evidence suggests that mutations in this gene impact microtubule stability, as well as other genes shown in this. However, what I'd like to tell you about today is our continued focus on SOD1, which it was the first gene described to cause ALS. And it's intriguing because it's a little bit of an outlier when it comes to this schematic. So SOD1 stands for superoxide dismutase. It is an enzyme that catalyzes this reaction. It basically destroys free superoxide radicals in the body. It was discovered more than 25 years ago. And at this point, there's more than, I believe, 150 genetic variants that have been described in the literature that can cause ALS. And importantly, a lot of these variants do not impede the enzymatic activity of this protein, suggesting that loss of function alone is not sufficient to drive disease. In fact, a series of studies done by a number of groups over the last 20 years have really shown that mutations in SOD1 drive degeneration through toxic gain of function effects, which are primarily driven by the fact that the protein tends to misfold. And we now know that even the wild type SOD1 protein has a natural tendency to misfold, but ALS-linked mutations increase this tendency to misfold significantly. And there are really at least two separate species of misfolded protein that have been characterized by researchers known to contribute to disease. The first one is soluble misfolded SOD1 protein, which is highly toxic both within the cells. Several groups have now shown that this can be secreted and taken up by other cells. And this soluble misfolded SOD1 protein can lead to ER-related stress, mitochondrial damage, and transport defects. It is currently unclear as to how exactly this misfolded protein leads to this defect, and that's something that I want to focus on today. However, I also want to mention that eventually this soluble protein becomes insoluble and forms this aggregates. There's a debate in the field about whether these aggregates are toxic or protective, but the fact of the matter is that postmortem tissue of ALS patients shows an abundance of these aggregates. Whether these represent survival neurons that have survived or not remains unclear. But what I briefly mentioned before is that there's a little bit of a black box in between the accumulation of misfolded SOD1 protein and all these degenerative dysfunctional pathways that have been shown in mouse models and other cellular models of disease. And our goal is to enlighten this black box and try and understand how a misfolded protein leads to these defects. We believe that it is important to use a physiological model system to ask this question, i.e. a model system which relies on physiological expression levels of the mutant protein. And a few years ago, we described the generation of the first patient-specific SOD1 ALS line. This was a female patient, about 43 years of age at the time of donating a little piece of their skin. This particular patient had very aggressive type of ALS and actually passed away a couple of years after participating in our study. So we generated patient-specific induced pluripotent stem cells, and we used gene editing to generate a corrected isogenic control patient line. This is just sequencing, Sanger sequencing, showing the presence of the heterozygous mutation, which encodes an A for V change in the amino acid sequence of the mutant protein. And this is just showing you that the gene editing was correct was successful in correcting this disease-associated variant. And we've done a number of quality control assays showing that there's no differences in copy number or mRNA levels of SOD1 between these two lines. Now, several years ago now, we published a study highlighting that you can utilize this model system to describe a series of pathways that are disrupted in motor neurons differentiated from these patient neurons relative to the isogenic controls. And these ranged from ER stress and the unfolded protein response, mitochondrial abnormalities, impaired mitochondrial trafficking up and down, processes hyperexcitability, and cytoskeletal dyshomeostasis. Although we showed that there is some level of misfolded SOD1 protein, we really did not address how the accumulation of this protein affects and leads to these downstream pathways. And an outstanding question is whether these are direct or indirect effects of downstream that occur downstream of accumulation of mutated misfolded SOD1. So a few years ago now, Kostas Choras, a postdoctoral fellow in my lab, decided to take on this challenge and try and shed some light on that black box between misfolded SOD1 protein and human neuronal dysfunction. So the first question that we wanted to ask was, what is the biochemical and conformational state of SOD1 protein in these patient motor neurons? And we employed a couple of different strategies to characterize the two different species of SOD1 protein that have been associated with toxicity. This is a schematic of the experiment that Kostas put together. So it takes us about 14 days to go from the induced pluripotent stem cells to post-mitotic motor neurons. And then we plate these cells, and for the context of this experiment, we kept them in culture for an additional 35 days for a total number of 50 days in culture. Now, throughout this process, Kostas isolated fractions from these motor neurons and worked closely with Jonathan Gilthorpe from the University of Umeå in Sweden, who's developed a rather accurate and specific ELISA-based approach to quantitate the amount of misfolded but soluble SOD1 protein. And these are the results of that assay here. Unsurprisingly, we found that the mutant motor neurons exhibit about 15 to 20-fold higher levels of misfolded but soluble SOD1 protein. But surprisingly, although there was some variability in the amount of misfolded soluble SOD1 protein appeared to be consistent across time in culture. Kostas also used a biochemical Western blot-based approach to characterize the amount of SOD1 protein that ends up in the insoluble fraction, as well as the amount of total SOD1 protein that ends up in the soluble fraction. This is just the Western blots exhibiting that analysis. But what I wanted to bring to your attention is that the mutant motor neurons as early as day 25 show signs of accumulating SOD1 protein in the insoluble fraction. This becomes a lot more pronounced as they mature and grow older in culture. Eventually, even the wild-type neurons accumulate this, but the patient motor neurons always accumulate more. This is quantified here. And what really we're showing here is that the patient motor neurons exhibit a progressive accumulation of insoluble SOD1 protein, which really becomes significant after about 30 days in culture. So now that we had a clear understanding of the dynamics of SOD1 protein, of the misfolded SOD1 protein in our culture system, we asked, well, how does the accumulation of this impact proteostasis? A long-standing hypothesis in the field has been that this misfolded SOD1 protein will impact global proteostasis. It will impact the ability of the proteome to function. And we looked at this in a couple of different ways. This is an assay looking at 20S proteasomal activity. We examined patient and isogenic control neurons on day 35 at a time point where we know there's sufficient amounts of soluble as well as insoluble SOD1 protein. And there was really no evidence of global proteasomal dysfunction in these cells at this time point. We also looked at the amount of ubiquitinated proteins using this two pull-down based approach in both isogenic control, in both mutant and isogenic control motor neurons. And we found, again, that at a time point when there's sufficient misfolded and insoluble SOD1 protein, there was no evidence for global impairment in proteasomal activity, as the amount of precipitated ubiquitin proteins was not significantly different. We also looked at the ubiquitin flux after blocking the proteasome using MG132. And while we show a significant accumulation of ubiquitinated proteins, there was really no difference between these genotypes. So having established that there was no global defect in proteasomal activity, we next wanted to get information at the single protein level and ask whether there were specific proteins that were degraded at different rates or that their turnover was affected as a result of the accumulation of misfolded protein. So to do this, we teamed up with Jeff Savas, who runs a world-class proteomics lab here at Northwestern, and employed an approach known as SILAC-based, SILAC, which stands for stable isotope labeling of amino acids. And what this really is, it's an approach which allows you to tag, to label proteins in the entire proteome with isotope tagged amino acids, and track their abundance as well as their turnover rates in the context of specific experiments. So what we did here is we literally fed our motor neuron cultures for 2 weeks with this SILAC-based media to label the entire proteome. And at the end of these 2 weeks, at the time point when we know misfolded SOD1 protein starts accumulating in the insoluble fraction, we switched the media and chased out the heavy proteome, excuse me, and perform mass spectrometry to quantify the abundance of older proteins as motor neurons mature. This is just some quality control experiments to show you that there were no statistically significant differences in the percent of the proteome that was labeled in the two genotypes. And I'm really showing you here that 75% of peptides were labeled at least 80% on day 30 for both genotypes. This is a quantification of the entire proteome, the labeled and unlabeled, and as expected, you see that as you chase out the proteome between days 30 and 51, the amount of proteins that are labeled disappear from the culture, and the amount of proteins that are newly synthesized appear in the culture. And there were really no differences between the mutant and the control motor neurons, suggesting that the pattern of proteome-wide degradation was similar between these two genotypes. However, when Kostas focused on proteins which were commonly labeled between both genotypes at each specific time points, he discovered that there was a specific set of proteins that seemed to be degraded at a slower rate relative to isogenic controls. Each dot here represents an individual protein in patient and isogenic control motor neurons at 3 different time points. And I hope you can appreciate that the mean intensity of the proteome is always significantly higher in the patient motor neurons, and there are specific proteins that are always more labeled in the patients than in controls. You can look at this data in a number of different ways. There's a total of about 150 proteins that exhibit a slower rate of degradation relative to controls. If you perform pathway enrichment analysis, you get things like the unfolded protein response and chaperone-mediated autophagy. But we really wanted to focus on the most robust changes. And what caught our attention was that there were 8 proteins which are consistently degraded at a slower rate relative to controls across all time points in this experiment. These are the proteins depicted here. And what really caught our attention again was that at the center of this was a protein known as VCP. And VCP caught our attention for a couple of reasons. Firstly, because it is a well-documented protein that plays a key role in proteostasis. And secondly, because mutations in VCP itself are known to cause ALS. Now, what this protein does is it identifies ubiquitinated substrates that are due to be degraded or turned over, and through an interaction with a number of cofactors, it extracts these ubiquitinated substrates and either presents them to the proteomes for degradation or releases them for turnover. And you can imagine that if the function of this protein is impaired, the turnover or degradation rates of a number of proteins would be impacted. So the first thing we wanted to do is validate our findings. So we repeated this exact same SILAC-based mass spectrometry experiment, but instead of looking at the entire proteome, we used an antibody against VCP to really pull down VCP itself and quantify using mass spec all the VCP peptides. This is the IP, and this is the quantification of the mass spectrometry peptides, showing you that indeed A4V motor neurons exhibit more older VCP peptides relative to controls. The next thing we wanted to do was to validate that this was not some artifact of this specific cell line. And as I showed you before, all the experiments were based on a patient line and an isogenic control corrected, but we wanted to ask whether the mutation in itself would be sufficient to induce this phenotype of slower degradation in VCP motor neurons. So for this, we took a wild-type line and we used CRISPR to introduce the SOD1 mutation, performed the series of laborsome experiments, and to cut a long story short, we found exactly the same thing, albeit with different dynamics, in that now the A4V motor neurons accumulated significantly more detergent and soluble SOD1 protein, and shortly after, they also exhibited accumulation of older VCP protein. So the next question is, does the accumulation of older VCP protein impair its function and contribute at all to mutant SOD1 toxicity? But I showed you earlier that there's no evidence for accumulation of ubiquitinated substrates in mutant motor neurons. So where does that leave us? So we scoured the literature, and what we found was two very intriguing things. First of all, the interactome of VCP, it seems to be exceptionally dynamic. It depends on the cell type and the stress of the cell. And second of all, mutations that are associated with multi-system proteinopathy, including ALS, do not lead to loss of function of VCP, but rather alter its function by altering its ability to interact with specific substrates. And in fact, loads of the variants are in protein-protein interaction domains. So inspired by this literature observation, Kostas decided to do another mass spectrometry experiment. Again, he labeled the proteome. Again, he pulled down VCP from wild type or patient motor neurons, and that's how does the interactome of VCP change in the presence of SOD1, of an SOD1 mutation. And to cut a long story short, what Kostas found was evidence for both gain of function interactions, shown here in the patient motor neurons, as well as evidence for loss of function interactions, shown here. Importantly, the overwhelming majority of the interactome was the same between mutant and SOD1 motor neurons, but there were significant differences. And there's, again, different ways you can analyze the data. This is looking at the data across time and looking at specific interactors in either mutant SOD1 or isogenic control motor neurons. And what I can tell you is that gain of function interactors are enriched for proteins involved in actin and chaperone proteins, what loss of function interactors are enriched for mitochondrial and translation initiation factors. Now, our kind of hypothesis is that this altered function of VCP might impede its ability to lead to regular turnover and recycling of proteins that play key roles in mitochondrial function, translation, cytoskeletal homeostasis, and so on and so forth. So the last couple of minutes, I want to talk about everything that I told you so far has been done in a culture dish. So is there any evidence of a crosstalk between mutant SOD1 and VCP in vivo? For this, we collaborated with Bob Cobb's group upstairs, who've been employing this beautiful worm models. And it turns out that when you overexpress mutant SOD1 protein in a worm, you impede its ability to move around the dish. This is a graphical representation of tracking of this movement. I hope you can appreciate that the wild-type worms freely move around in the dish, whereas the mutant SOD1 worms are limited in their ability to move around. This is a quantification of another metric, the average speed at which these worms move. And you can see that when you overexpress mutant SOD1, there's a significant reduction in the average speed of the worms. When you knock out VCP you get a significant reduction. But importantly, when you manipulate VCP in the context of SOD1, the SOD1 background, and specifically when you knock it down, the phenotype becomes significantly worse. But most importantly, when you overexpress VCP in the context of this mutant SOD1 background, you can completely rescue this defect, suggesting that there is a functional genetic interaction between VCP and SOD1 in an intact nervous system. Last but not least, we went back to postmortem SOD1 patient tissue, and these were samples that were graciously donated by Ray Roos to us. And you can see that in spinal cord of control tissue, there's no evidence of SOD1 or VCP aggregation. However, when you look specifically at an SOD1A4V patient, there's an abundance of SOD1 aggregates shown here and here. And intriguingly, there's evidence for an abundance of VCP accumulation, which is likely aggregation in the same motor neurons. So in summary, we've begun to illuminate this black box between mutant SOD1 and neuronal degeneration, and have highlighted a previously unappreciated functional interaction between two genetic causes of ALS, SOD1 and VCP. And we hypothesize that altered VCP dysfunction is a major contributor to the dysfunction of mutant SOD1 motor neuron and leads to degeneration. I would also like to point out that while we've illuminated this black box, at the same time we've created some other black boxes in between here. We do not entirely understand why VCP exhibits an altered function in the presence of SOD1 protein, but we're currently investigating that. Now, why is this important? Well, if you think about the overall ALS population, as I mentioned, the overwhelming majority is of a sporadic nature. These different color people here represent different familial or genetic subtypes of ALS. And our work has clearly shown that there is a potential interaction between these patients here that suffer from SOD1 ALS and these patients here that suffer from VCP ALS. This is important because a potential therapeutic, either targeting VCP, which could be relevant for VCP individuals, could have some therapeutic implications for SOD1 patients. Last but not least, an outstanding question which we're always interested in addressing is whether does any of these have any relevant sporadic patients? And this is something that we're also investigating. So I'd like to finish there, and I've highlighted our collaborators throughout the talk. Kostas has done a phenomenal job on this. Kevin Smith and Beth Daly have contributed to this. These are all my lab members. Again, I've highlighted our numerous collaborators and our funding agencies, particularly the New York Stem Cell Foundation and the Les Turner and Les Foundation, as well as the NIH. Big thank you for supporting our work, as well as the patients that have contributed samples that have made our studies possible. So thank you, and I'll be happy to take any questions.
Thank you, Evangelos. That's really very exciting work. And unfortunately, we're sort of butting up against the time limits. I want to make sure that everybody gets an opportunity to complete their study. So I have, as I may have mentioned before, I have a list of questions that have been asked, and I'm going to collect them and I will give them to you to discuss offline or at the end of the meeting. So thank you very much, Evangelos.